Welcome to our project,
PATRONUM.
To give you a better insight of what exactly root rot is, root rot disease is a soil-borne disease caused by a family of fungi called Fusarium. It severely affects the yield of soybean, especially in China, Heilongjiang. It causes around 50,000 ton yield loss of soybean annually. And as soybean is used widely in industrial production, this decline will cause an international consequence. As you can see on the diagram, root rot is causing problems around the world. It's causing a decline in crop yield to up to 80%, especially in Nigeria and Brazil. Traditional solutions include crop rotation, which is slow acting and require long time long time practical experience, and it will be relatively inefficient on late to mid stage of root rot.
So chemical control, which commonly relies on the application of chemical agents, is not a method employed for root rot management. However, it has two notable drawbacks. First, chemical control entails risk of pesticide residues and development of pathogen resistance. Second, chemical use can lead to environmental pollution. Compared with traditional methods, microbial fertilizers offer advantages like ecosystem protection and green planting needs. However, they have weakness that their biological agents have weak efficacy. Thus, there's an urgent need to optimize the action mechanisms of biocontrol agents and develop novel functional strains with enhanced performance. Our solution includes the two following methods of fighting root rot disease: chitinase and β-amyrin. Chitinase is used to break down chitin in cell walls, while β-amyrin hinders chitin production in the first place. For the first method, the chitinase-expressing strain degrades the existing chitin in cell walls, destroying mature pathogens.
To optimize efficiency of the chitinase secretion, our framework comprises 3 steps. 1, we develop a pipeline for selecting the optimal signal peptide, and the second is the wet lab verification using SFGFP. And then is the verification of chitinase secretion and activity effect. Here's the overall workflow we designed for selecting the optional secretion signal peptide. The first step is that we collected a set of signal peptide sequences from the database and fused each candidate with a specific linker and the target protein. Then we also use computational tools to evaluate secretion potential. You can see here, signal P confirms the secretion pathway and cleavage site. TMHMM eliminates protein with multiple transmembrane helices. And allows deep block used as the assistant of validation. So 3 tools here allow us to effectively have a screen of our candidates. So we also designed an energy rating system. N region is the positive charge, and H region is the hydrophobicity and length. And last, C region is the AXA motive site proximity, or weighted. In addition, signal P score and cleavage site probability also help us to generate a ranked prediction of efficiency of the secretion peptide.
Oops. After that, we select 6 potential signal peptides with high-ranking one and low-ranking one. This strategy allows us to verify our signal peptides selecting system, while allows us to choose the optimal signal peptide with a minimum cost. Next, we use SFGIP to decide the effectiveness of our signal peptide. We consort a gene containing our potential signal peptide and SFGIP. And then we clone that gene into a vector of P1828A for inducible expression with IPTG. And SFGIP allows us to visualize the fact of secretion ability. Higher light intensity would indicate better ability of secretion. We observe biofluorescence under blue light, and this is a quantitative measurement about secretion ability. The higher the light intensity is, the better the secretion ability is. We also did a quantitative research by microplate reader, where the relative light intensity of different signal peptides are measured. From the diagram, you can see that OMPA works the best, which was consistent with the model prediction result we made. After that, we also choose our optimal chitinase. We fused all these potential chitinases with OMPA, which was the optimal signal peptide we found before that. We construct a gene consists of chitinase and OMPA. Then we clone that gene to PT28 again for IPTG inducible expression. And the superintendent was analyzed using electrophoresis. It turns out that the BL key and BS key are successfully expressed and secreted. To detect the biological activity, we actually did a colloidal chitin plate assay. We put our colony onto our chitin plate, and a hydrolytic zone around our colony indicates that BL key and BS key are both active, both have biological activity.
Our second method was to produce β-amyrin, which is significant in inhibiting cell wall synthesis via binding to chitin synthase in Fusarium fungi. Here is our framework. First, we constructed the β-amyrin-producing E. coli strains. Then we detected the β-amyrin production. And finally, we saw the need to introduce our exogenous MVA pathway to increase β-amyrin production. Previous research showed that there's already an endogenous MEP pathway in E. coli to produce FPP from pyruvate. And from FPP, we can further produce beta-amyrin. So we inserted these three genes into our recombined plasmid PTYT, and then we introduced our plasmid into DH5 alpha cells. And here you can see the successful colonies of DH5 alpha cells. After fermentation culture and product extraction, we detected the production of beta-amyrin using high-performance LC mass. Here are our results. On the left, you can see the characteristic molar mass of beta-amyrin detected, and we allowed an error of less than 10 ppm. On the right, you can see that at a retention time of roughly 12.2 minutes, we have both a peak in the PTYT group and the beta-amyrin standard group, showing that we indeed secreted beta-amyrin. However, the endogenous MEP pathway only meets E. coli's basic physiological needs, so we did not produce that much beta-amyrin. To produce more beta-amyrin, we researched and found that in yeast, there is the exogenous MVA pathway. So here, we introduced the plasmids PMEV-T and PMBIS for our exogenous MVA pathway. And you can see that again, we get successful colonies of DH5 alpha cells. After introducing the exogenous MVA pathway, we ran LC-mass again, and it supported our hypothesis. As you can see right on the graph, the 1 plasmid group has significantly less beta-amyrin production than the 3 plasmid group. So by introducing the exogenous MVA pathway, we successfully increased beta-amyrin yields. And to test the antagonistic effect of beta-amyrin, ideally we would use Fusarium oxysporum directly in our tests. However, it is in risk group 2, so high school teams cannot use it. So we need to select an alternative organism with chitin synthase that is similar structurally to Fusarium's, because beta-amyrin binds to chitin synthase. So we developed our biosafety tool. We first got all the whitelist of all the organisms we can use. We also got the core.fasta file for the Fusarium chitin synthase. Then we downloaded all the whitelist organisms' candidate homolog sequences from UniProt, and then we did homology screening and pocket mapping. We looked at shape overlap, side chain chemistry, RMSD, which is root mean square distance, and also PLDTT quality control. Finally, we found that S. cerevisiae, which is yeast's chitin synthase 2 is the most structurally and functionally similar to Fusarium's chitin synthase 3. So that's what we chose for our antagonism experiments. And in 2022, GEMS Taiwan also tried this. However, their results were very underwhelming. Very little amounts of yeast died. We suspect this is because yeast only has about 1 to 2% of chitin in its cell wall. So even if all the chitin was gone, it would not have that huge of an effect on yeast. That's why we pre-treated yeast with snailase, because snailase degrades the other parts of the cell wall so that the cell wall is more dependent on the chitin scaffold, so our chitin antagonists can have an amplified effect on yeast. For our first antagonistic assay, we used fluorescence. We obtained a red fluorescent protein producing yeast strain from Tsinghua M. And when the yeast dies, RFP will be released outside of the cell so we can detect that fluorescence. For our quantitative flow cytometry antagonistic assay, once the yeast dies, PI dye will enter the nucleus and bind to the DNA, and we can also measure that for our antagonism tests.
Specifically, we use an RFP expressing Cerevisiae strain, and fluorescence intensity measured by microplate reader served as a quantitative indicator of the degree of cell rupture. The experimental results showed that fluorescence intensity increment of the group pretreated with snailase was significantly increased compared with the original experimental step group. This indicates that snailase pretreatment greatly improves the detection system. It sensitizes the otherwise unresponsive Cerevisiae to chitin, thereby enhancing the system's effectiveness. One disadvantage of the simple method is that fluorescence intensity may be affected by other fluorescent substances into supernatant. In order to gain a more accurate picture, we use the second method, which is a quantitative method, pi staining with flow cytometry. We apply snailase pretreatment and then use pi stain to label dead cells. The results were more accurate, which means both chitinase and β-amyrin and snailase pretreatment resulted in the death of 65.8% of Cerevisiae cells. This shows a synergistic antagonistic effect between chitinase and β-amyrin. This was a massive success, and we found a way to effectively destroy root rot disease.
All right, with all that progress, human practice kept us on target. We organized our work across 6 sections: product and user end, technical end, academic exchange, safety and regulation end, production end, and product form experimentation. In Liantang, farmers prioritized safe, residue-free pest control methods, where chemicals were fast but toxic, while biological methods were slow but safe. This inspired our goal, a biological inoculant that enhanced chitinase for a stronger antagonistic expression. To turn ideas into reality, we interviewed Professor Wang Jinbing, who emphasizing signal peptide selection. We built a computer model screening over 2,000 peptides and tested the top candidates with chitinase from multiple genera. Professor Li Yan's insight on a synergistic dual pathway was pivotal, showing that beta-amyrin inhibits chitin synthase and making it an ideal partner for chitinase. At the CCIC conference, we achieved 2 milestones. First, being recognized as the top 10 team, as the only high school team. And second, securing a vital collaboration with the Tsinghua M team, who provided Cerevisiae to accelerate our antagonistic experiment. Lawyer Zhang clarified the commercialization liability and international regulatory gaps and ARG risks. BlueFuz's recommendation solidified our powder inoculant as the final product form. And in the future, we'll continue to investigate our products, in the future, we'll continue to investigate our synergistic dual strategy in B. subtilis, and through chitin plate hydrolysis experiment and Western blotting. So in the future, when pathogens roll in like storm clouds, we hope farmers will lift our product.
Let's give another round of applause to Team Uprize-I. So please go off the stage from this side and follow the green carpet, please.